La primera plática va a ser sobre el papel de los microorganismos en la calidad de la carne, en el deterioro y calidad, deterioro que están relacionados y en la inocuidad. Es la doctora Linda Saucier, que es profesora investigadora en el Departamento de Ciencia Animal de la Universidad Laval, que es miembro del Instituto de Nutrición y Alimentos Funcionales en Canadá. Hizo su licenciatura en el 86 y la maestría en el 90, ambas en el área de Ciencia y Tecnología de Alimentos, justo ahí en la Universidad Laval, y posteriormente hizo su doctorado en la Universidad de Alberta en el año 97 en el tema de regulación y producción de bacteriocinas por Carnobacterium piscicola. Después de su doctorado trabajó por más de 6 años para Agriculture and Agri-Food Canada, obteniendo reconocimientos internacionales como el que otorga el Secretariado Internacional de la Carne en el área de Ciencia y Tecnología de la Carne, por un ensayo que escribió titulado Inocuidad de la carne, retos futuros, que fue publicado en 1999, y en 2016 recibió el premio John Sophos al artículo de revisión más citado en la revista Journal of Food Protection. Entonces, bienvenida a la doctora Linda Saucier. Ahorita le vamos a dar la palabra. Esperamos que la presentación sea del interés de ustedes.
Es un placer, pero mi español no está muy bueno. Entonces, lo presentará en inglés. Lo siento. Okay. The first thing I want to address, we're talking about...
Díganle que se pare, por favor.
So people do have to have the means to be able to buy the food. They have to be able to utilize it.
¿Podría permitirnos tantito, por favor?
Linda, es Claudia, ¿cómo estás?
¿Cómo está?
They are having issues with the volume because we cannot hear you, so can you wait a little bit while they'll fix it?
Okay.
Pero no se escucha.
Afuera donde no se... A ver, dame para lo más. Linda, let's make a test. Can you say 1, 2, 3, 1, 2, 3 to check?
1, 2, 3, 1, 2, 3, check.
No, no se oye.
Se oye, pero muy bajito.
No, it's not working properly yet. Ya le digo que pruebe. Linda, please again, 1, 2, 3, 1, 2, 3.
1, 2, 3, 1, 2, 3, test.
Okay. Ahora sí, ¿no?
Sí, muy bien.
Perfect. Go ahead, Linda.
Okay, so we were talking about food sustainability. So I was referring to utilization. People have to be able to use the food that we provide for. Let's say you give a grower some crops and this land is not suitable for that crop, so then it's not worth it. So in terms of stability, it has to be available all year long. So you cannot have famine and then a lot of food, so you have to have it all year long. Unfortunately, a third of the food we produce for human consumption is either lost or waste globally. So if we do manage to improve safety and spoilage, and a lot of it is caused by microorganisms, we should be able to improve and reduce that quantity of food that is lost every year and that we do put energy in producing. With respect to meat, it's 20% of the meat we produce that is not getting into the market. So that's equivalent to 75 million cows that we basically produce every year and are not consumed. So we want to do better, we want to improve our food supply, but we have to be careful on the way we do it, because too often perception is deceiving. Now we know that animal welfare is important to secure meat quality, but we have to have some science-based analysis behind it, otherwise it's not going to work. Just referring here to some free-range animal. A lot of people think that it's better for the animal, but one consequences that can happen is that those animals that are free-range are now getting more contaminated with parasites. So we think that it's nice to have them, gaze outside, yes, but that means you have to better manage your land, better manage your water source, control vermin. So it's not just because they feel better that it's all right. You have to do the science behind it and make sure that what you do not deteriorate your quality or your safety of your product just because you think that it's good for the animal. So animal welfare is important for meat quality, but we have to be careful how we manage it, and it should be on science-based and not perception. Another example to outline what I'm trying to say is, if you look at luncheon meat, okay, you can have it in the retail, prepared and sliced in the shop, or be prepared industrially. Now the question is, which one is safer? The retail one that is casual and you have the person preparing for you, or is it industrial? Well, the prevalence of Listeria monocytogenes is 8 times greater at the retail compared to a luncheon meat that is prepared industrially. So it's not because it's nice and bucolic that it's necessarily good. We have to do the science behind it and in order to control microbial spoilage. Now, the way we've been doing things for many years is to look at the total microflora. And we've been reducing total microflora because doing so, we also reduce the risk of exposition to pathogens. And that's good. However, what you create as well at the same time is also an environment exempt of competition for organisms that contaminate your food post-treatment. So you basically create what I call a microbial void. So there's no more microorganism, and then there's a hole there. So that means that if you have contaminants, there's no competition. They can get there, contaminate the food, and be able to thrive. You basically have created a Club Med or a buffet for those contaminants and are able now to colonize that void that you create. So it's nice to be able to reduce the microflora, but that means you will have to control as well the contamination that may occur afterwards. So maybe we have to rethink the way we're doing or create or look at new ways to secure our food supply. What I'm suggesting is maybe we are at a point that we should rather be doing microflora management rather than, eliminating everything. And I mean by that, having the microorganisms that you want, at the time that you want, and at the quantity that you want. Because our food is a source of nutrients for all microorganisms. So we have to look at it in this perspective. So what we've been doing so far is really looking at reducing the total microflora, but now I'm suggesting that maybe it's time to find a means to eliminate the undesirable microorganism and leave the desirable one behind. So it means rather than having a bazooka approach, it's rather having a sniper approach, because we know our food, if they are good, I mean, microorganisms are likely to contaminate them. So we're getting into competition of those microorganisms, and we're looking at better ways to control the whole environment. Best example of this is modified atmosphere packaging. That's exactly what we've been doing. In this case, when you take meat and you package it aerobically, like in your styrofoam tray with your absorbent pad, your meat and your saran wrap on top of it, you got air. So Pseudomonas thrives on that condition when you are in refrigeration temperature. They grow fast, and soon enough, when they are about 10 to the 7th, they produce slime and bad odors because they use the amino acid. They decarboxylate them and produce compounds like putrescin and cadaverine that smells very bad. So that's what happened in aerobic condition. Now, if you take the same meat and you put them into a vacuum packaging or a environment where there's lots of CO2, no oxygen, then it's the lactic acid bacteria that prevails. Because they tolerate more CO2, they grow slower, at a slower pace, so it takes longer for them to actually spoil the meat. So it's only when they get to 10 to the 8, 10 to the 9, that the meat then is discarded or judged as spoiled. So you basically then go from a shelf life of a few days to a few weeks, just by changing the atmosphere, the packaging you're using. You're not using any chemicals or irradiation. You just control the microflora. So that's the first example of what I'm saying, microflora management, not producing it, controlling it, having the one that we want, at the quantity that we want, at the time that we want. Now, let's go ahead. Here in Canada and a lot of places, we've been cleaning and we've been improving a lot in what we've been doing in our plants to improve the salubrity and the inocuity of the food we produce. But unfortunately, when we talk about meat, everything is processed at 10 degrees Celsius max. And most of my processors are more around 1 and 4. So the only microorganism that is able to thrive in that environment, or one of the few, is Listeria monocytogenes. So Listeria monocytogenes has been a problem in meat plants because in the drain, notably, you can have easily something like 10 to the 5 log at certain times. And we know that drains in food plants will never be sterile. You're always going to have water and organic matter, so a lot of microorganisms can grow there. So what we've been trying to do is to look at a consortium of bacteria that is normally produced by industrial processors to control bad odors in drains and grease traps. So it's a commercial product that is used in restoration a lot. And it contains something like 6 to 10 to 7 log of cell that you can dilute 1 in 1,000 to be used. So what we did, we looked at 4 of those commercial products that are constituted of a consortium of organisms, and see if they are able to exert a competition against Listeria monocytogenes. So because meat plant or processing are usually at 10 degrees Celsius and less, we did the experiment at 10, and this is how it happens. Ali. So you have here at the bottom how the consortium grew in brain-heart infusion at 10 degrees Celsius. It grows slowly, but it does grow. Now, if you look at the 2 top one, it's Listeria innocua. You can see that at 10 degrees Celsius, it thrives and grows, no problem. But when you put the 2 of them together, yes, the consortium put a pressure on Listeria monocytogenes, and they're not able to grow as much. And yet, if you look at the 2 bottom line, you can see that they don't grow much. So maybe there's room for improvement to find organisms that are, consortium that would be working better at refrigeration temperature in order to put a heavier pressure on the stereomonocytogenes. But here we have some of a proof of principle for, products that are used to maintain the good quality of grease trap in restaurants and so on, for odors mainly. They're able to put a pressure on the stereomonocytogenes so that it doesn't grow to the same level. So again, that's an example that maybe rather than trying to remove everything, we should rather be looking at the right microorganism to grow and take over that environment. Now, everybody knows about probiotics. I'm sure if you're a microbiologist, you've been working in the food industry, and if you are, animal grower, you know that probiotics does exist either for human and animals. There's commercial product available from the feed company. Their efficacy has been demonstrated on the health aspect of human or animal. So that has been demonstrated, and we use them. Now, a couple of years ago, I had a project with this friend of mine, Colin Gill, and Colin was looking at the prevalence of Mycobacterium avium, the subspecies paratuberculosis, which is the agent of Johne's disease in cattle. And we were looking at plants, if they were on the carcass. Colin was taking care of the analysis out west in Canada, where they grow more beef as meat, and in Quebec it's more cattle for dairy cattle. So he said, if we want to do a good sampling, you should be doing the dairy one. So I said, okay, I can give you a hand, no trouble. But I said, Colin, there's not that much Mycobacterium on, in the field. The cows, they don't live that long before we send them to cow. And so I said, are you sure we're going to find some? And I will always remember what he told me. He said, Linda, if it's in the feces and we work well, we should find it on the carcass. And sure enough, we did find few, but on many carcasses. So that has always stuck with me. And then I'm proposing, if we feed good bacteria, and the same principle, will we be able to influence the microbiology of the carcass? So we know we can give probiotic to animals and humans, and it's legal, and you have commercial product, but that was for the health effect. But can we have also a beneficial effect on the microbiology of the carcass if we give the animals the right bacteria, the bacteria that we want to find on the carcass, rather than letting the Enterobacteriaceae take over? So that was the idea, or the hypothesis at the time, that what we did with Colin sort of a trigger. So what we did, we supplemented the feed of rabbit. Well, rabbit, you say, why rabbit? Well, rabbit is popular here in Canada, not as much as the regular one like chicken and pork and so on, but we do have a certain production. And so you get your slaughter weight in a couple of weeks, like 4 or 5 weeks, you just slaughter weight and you can go to the slaughterhouse. So what we did, we supplement the feed of those rabbits with a protective culture that is that you can use in Canada. It's homologated for ready-to-eat meat to control Listeria monocytogenes. It contains a bacterium, Carnobacterium maltoaromaticum, and this is a spin-off from the University of Alberta, from Dr. Stiles and Dr. Macmillan's laboratory. And this organism produced 3 bacteriocins that are active against Listeria. So what we did, we separated rabbits in 2 groups. They were raised in commercial cages, but in a research facility where we were able to isolate them with, biosecurity to make sure the 2 groups won't contaminate one another. And we look at the prevalence of Carnobacterium maltoaromaticum on the meat and the carcass. So I won't go into all the details of it, but I'll show you one result that is, to me, very determinating that our hypothesis that if you feed the good bacteria, you can have a positive effect on the carcass is this one. So from those rabbits, we use the meat and we ground it and we inoculate it with Listeria monocytogenes, and we stored the meat either at 4 or 10 degrees Celsius. So if you look at the two graphs, you have the ground meat stored under aerobic condition and at the bottom under anaerobic condition. In the top one, you see 10 degrees Celsius would be the two first line, and then 4 degrees Celsius, the two bottom one, and that's the same thing for anaerobic condition. Well, at the end of the storage period, we were able to reduce the growth of Listeria monocytogenes by 2 log. So again, we didn't add any chemicals, we didn't irradiate it or whatever. We just gave to the rabbit a good bacteria, and yet in the end, when we took the meat from those rabbits and inoculated with Listeria monocytogenes, we were able to get lower count of Listeria monocytogenes. Though it's maybe not just the fact that they ate it and it got into their gastrointestinal tract and the feces. There's probably some contamination as well of the fur and the hide. But in all of it, because we put a good bacteria in the system, we were able to improve the control of Listeria monocytogenes. So again, not sterilizing things, but just adding the bacteria that we want, the good ones, in order to improve our microbial control. Now I'm getting a little bit more, I don't know if I'm good on time, I know I have 30 minutes. Just want to touch base on a few other aspects with respect to how we evaluate the efficacy of antimicrobial system. So far, traditionally, we've been using cell count to establish log reduction in order to determine if an antimicrobial system works. So the cell needs to grow to be detected, but if the cell is injured, it won't make a colony. So you're likely overestimating the efficacy of your antimicrobial system when you have injured cell present. If you rely the efficacy, or you determine your efficacy using only cell count, because the bacteria, if they're injured, they won't grow. Especially on selective media, where you add again other antimicrobial to be able to be selective. And if they're injured, you put a pressure that makes them difficult to grow and form a colony. So again, we are overestimating the efficacy of our antimicrobial system. So the cell really needs to get over the stress and to recover in order to be able to grow and form a colony. And resuscitation is not always optimal or possible. Okay, now I'll make a difference between persistent and persisters, because often they are mixed up. Persistent is the cell you have in your plant that is always giving you a problem. You think you have the control, and then it's coming back. And then you do other things, you do other change, and then it's always coming back. But the persisters is something totally different. They are associated with the use of antibiotics. And those persister cells, which is particular, they do have a toxin antitoxin system. And the persister do not grow like mutant that acquire antibiotic resistance. Like the ones that are resistant, acquired a resistance, do grow. But persisters, they don't grow. What happens is that they just stay there in dormancy, and they resume growth when the antibiotic is removed, or when the concentration is low enough. So this is how it works. And just to show how when cells are in dormancy or not capable of growing, this disturbs the way we evaluate the efficacy of our antimicrobial system. So in this case, you have an antitoxin linked to a toxin. And there is many systems, like in E. coli, there's something like 26 systems like that. So when you add the antibiotic, the antitoxin, which is not very stable, gets degraded by protease, and then that releases the toxin that puts the cell to sleep. So it reduces the protomotive force, reduces the level of APT that is produced, so that the cell is just metabolically slower and put in dormancy. So that kind of cell, you cannot count them because they're not growing. Now, another type of cells that is causing us problem is the viable but not culturable cell. The first example that was demonstrated about this state of a viable but not culturable state was with Vibrio vilnificus at low temperature. So in nature, basically when the cold temperature arrives, it dies off in the aquatic environment. Okay? Those cells that are still viable but not culturable in laboratory, they are more resistant to stress, and they remain pathogenic, although you need more cells to cause the disease because they're not culturable, so their metabolism is not very fast, not very active. It's slowed down, the metabolism is slowed down, and they resuscitate related to quorum sensing. So when they feel that growth can be restored, then boom, they go back and then they become culturable again. Okay, so again, when cells are put in situation where they are not culturable, for whatever reason, because the conditions are not right for them to grow, you just won't be able to count them and estimate them. There's all sorts of cells dormancy mechanism as well, like stationary phase is one. Cells that are in stationary phase because the nutrients have been exhausted become more resistant to all sorts of stress, and growth slow down slowly. There are certain spores that are particularly dormant. Spores is a form of dormancy for those spores former. It just stays, it's very stable in the environment, very tough to destroy. So it's another form of cells that are able to sustain rush environments. It's a bit like our bears. It's in dormancy all winter long, and then when springs arrive, it comes out and it's able to strive again. But all its metabolism is geared, so it's very similar. So all those states of dormancy and when they are not able to grow, then they're off the radar and we cannot count them. So we overestimate the efficacy of our antimicrobial system we're trying to study. Now, I'll go a little bit with hurdle technology to get me to where I want with the bacterial stress response. So you're all familiar with this concept because you're working in the food industry. So if you use, let's say, an antimicrobial system A, certain cells will be sensitive. But if you have to control cell 2, well, you need a higher concentration, and cell 3, an even higher concentration. Now, if it is acid, well, when you get to try to control cell number 3, it can be pretty acidic, so then the taste will be, affected and so on. So what we've been doing, and doing it well, is to use 2 antimicrobial systems in order to create a hurdle. So then that's that's as effective, and the impact on the food is lower because the 2 of them together are less stringent. But what happened to 3 and 2 if they are exposed to A? This concentration does not kill them, but it might actually induce all sorts of adaptation mechanism within the cell. So that's maybe something in using the hurdle technology that we've been underestimating, or that got forgotten in the process. So what we did, the thing is that when you look at cells 2 or 3, they're probably going to improve their resistance, and there's numerous examples in the literature that you can improve the resistance of a cell by exposing them to lower concentration or sublethal concentration. Okay, and that induces a bacterial stress response, and stress protein have been identified. Certain protein are reduced, others are downregulated. So there's a whole metabolism that gets into play. But what we don't know with respect to food and microbial spoilage or microbial safety and control is that the role, the mode of action, and the impact of those stress protein on the efficacy of antimicrobial system used in food are poorly understood. Because right now, a lot of those systems have been used in laboratory. We know they exist, we know how to induce them, we know which proteins are involved. But when you are in a complex environment like food, it's not the same thing. So there's still lots to be done on that field in better understanding that bacterial stress response in our food condition. Okay? This bacterial stress response can be of two kinds, general or specific, meaning that certain stress will induce a response that will make the cell more resistant to all sorts of other antimicrobial systems. And there's others that it's only one way, like you increase the resistance and it's only towards that antimicrobial system. So that's how it plays in the metabolism of microorganisms. So what we try to do here is we use two antimicrobial systems, salt, like an osmotic shock, and an acid shock. So we use these two, the salt stress and the acid stress, in different order. So in the first panel, you see salt was added, or the osmotic shock, the cell were treated at last with the osmotic shock with high salt concentration. And on the other panel is the reverse. Salt was used first and acid last. Okay? So those two were used at sublethal concentration. So when you use the acid first and then the salt last, you have a nice control of the optical density, so your cell don't grow as much. When you look at the other panel, when you put the salt first, the salt stress, the osmotic stress first, and then the acidic stress, well, the cells are not as controlled. The growth is better than in the first panel when the salt stress or the osmotic stress was done at last. And one of the reasons is that salt stress or osmotic stress generate or induced a general stress response towards a lot or several type of antimicrobial system. So it's completely different. It's the same stress, just applied to the cell in a different order, and yet you get a different response in terms of their growth. So you can see here, it's a picture of a micrograph, electron micrograph. You can tell that when you have the control, the acid stress and the osmotic stress, that the mode of action is totally different. I mean, the cells react totally differently. Yet, what we've seen is that the order in which you apply them has an impact. We know that when we use the two sublethal stress together, there is no growth. And that's why the hurdle technology works. So we have to be careful. When you're using hurdle technology, make sure you are using them together, because if you separate them, then you might induce some resistance. You improve the ability of the cell to adapt and resist. So we have to be careful and make sure that we do some evaluation, shelf life, etc., to be sure that it's worked properly. And that leads me to the mode of action, because if we're trying to use antimicrobial system and they're all working in the same way, like all affecting the cell membrane or the protein synthesis or whatever, you're in the same path. So maybe the question is, or the hypothesis is, we should put together antimicrobial systems that have different mode of action in order to be more effective. So this has to be in our mind when we're designing processes and processing line, because maybe the mode of action we're using in different system will influence. And we have lots of sequence when we do process food. Like if you are evaporating or drying milk, you first have to heat the milk slowly before you put them into the evaporator and before it gets into the dryer. So the big question is, are you adapting along the process yourself to be more resistant to the heat treatment you're trying to apply? So this is something we have to jiggle with when we talk about our processing. Now, one of the best example I can give you on that front, and we all use heat treatment to control microorganisms. This has been used for years and years. How does it work? So in the cell, the control cell at 37, like we're talking about E. coli here, we have the RPOH RNA in the cell. And this is the sigma factor that induces all the proteins necessary to resist to heat treatment. So it's already present in the cell. And DNA K, which is a chaperone, that's in blue, helps, maintain the protein in the right conformation, helps degrading the ones that are no longer, and it's linked to sigma 32, so the protein that is related to RPOH in the cell when it's in this, control state, growing nicely. So that's how it is with the control cell. Now you apply heat, what happened? The mRNA of RPOH, unfold, and then all the sequence being produced, you have sigma 32 that is produced, and then it's going to go and induce the promoter and gets all the protein that you need to resist to your heat treatment. Now, when you look at DNA-K, it's linked to a sigma 32 when the cell is in, growing in the control state. When proteins are denatured by the heat treatment, it has more affinity towards denatured protein than it has towards sigma 32. So again, sigma 32 can leave and activate the promoter and the proteins that goes out. So already in the cell in its control state, it has everything it needs to start the reaction to be able to adapt to a heat treatment. So that's how fine and refined the system can be. So what we've been doing is looking at DNA-K precisely during heat treatment and see how it goes, in treatments that are used in the food industry. And why DNA-K? Because it was easier to track with an ELISA test because it can get up to 13%, of the protein content when the cells are stressed by heat. So it's one that you can easily follow. So this is what the results of it. So we use different temperature, 50 degrees Celsius and 55, and you see on the left panel on your side the DNA-K molecules that are produced. So you can tell now that using different pasteurization value, although you have the same pasteurization value and that the treatment is supposed to be equivalent according to the theory or the principle of a pasteurization value, it's not the same. It's not the same quantity of DNA-K that is produced, whether you are at 50 or 55. And if you look at the right, at the cell concentration, you see again that the cell concentration at 50 and 55 are not the same. Although, again, because of the pasteurization value principle, that a pasteurization value of X, whether it's done with different temperatures, is supposed to be equivalent, doesn't work here. When you are above 60, no problem. No more DNA-K, you have a good cell control, no problem. When you are at 50, it takes 5 hours to reduce count of 1.5 logs. So that's not very effective. So here we clearly see, because of the DNA-K improving or increasing in the cell, that you are actually your treatment is not effective. The cell is actually adapting and trying to resist to the condition you are imposing on them. So now, the big question is, if you have more DNA-K, are you more resistant to a lethal treatment? So this is what we've done here. So you have two treatments. The one at a pasteurization value of 3, and then on the right at 5. So the 5 is lethal, the 3 is not lethal. If you look at the cells, once they've been treated at a pasteurization value of 3, and then you let them grow, at the optimal temperature, you bring them back to where they like to be, where you look at the DNA-K concentration, and it does increase as the cell gain growth again. So remember that 12, where the concentration of DNA-K is very high. Now, with the pasteurization value of 5, no DNA-K, no cell growth, even if you let the cell grow for 24 hours, doesn't move. So they're basically killed. So that's a lethal treatment. So now what happens when we take those that have a high concentration of DNA-K and we impose on them a lethal treatment? So this is the next one. So here we have control cell, that are at a level of 8.2, and then you apply the lethal treatment. After the treatment, you are under the detection level, and even if you let the cell recuperate for 24 hours, you get nothing. Now, we use the cell on the other panel 12 hours after recuperation in a sublethal treatment, So the stress cell, they were same log magnitude. We treat them with the sublethal treatment, where we know that after 12 hours they get a lot of DNAK, and then we apply the lethal treatment. Right after the treatment, we don't see a thing. But let them grow 24 hours, you're at 7.36 log. So cells have regained growth. So that basically says that those cells that were stressed, treated lethally, and then let recuperate, those cells, those daughter cells, remember the stress that their mother had before, because they were able to gain growth, to be able to go back to growth, because they've been stressed enough, just enough, to remember and to be able to regrow. So again, okay, how the state of the cells, the condition we've put their metabolism in, might influence how they're going to react to something when we try to kill them afterwards. So we have to remember this induction of stress is likely to stimulate a better response by the cell and make them more difficult to control. So again, that says that doing shelf life studies is important. You cannot just say, oh, we've treated this thing, we just changed this little thing, it's no big deal. No. When you change your process, you really have to go through shelf life studies to make sure you're not overlooking something important, that you have not induced a bacterial stress response that would make your treatment less effective. Now, if we look at other treatment, and we did some microarray analysis when we push, the cells in order to try to identify where the adaptation stopped. By doing this microarray analysis, we were hoping to find where we get to near-death physiology, when the process begins to be very effective and that we leave the adaptation capacity of the cell. So in the middle, you see 71 degrees Celsius. It's the temperature we use to cook ground meat to make it safe. We know it works. And you have other temperature. So the reference one is 37, and you have 58, 60 at two different pasteurization value. So when you're at 58, you manage to get some cell growth if you let them recuperate. But at 58, pasteurization value of 3, and at 60, you get nothing. But when you get at 70 and you look at this chart, you can see that the cells at 71 that have been treated at 71 degrees Celsius are closer in their microarray analysis to the control than they are to the treated one, the one at 60 and 78 at a PV of 3. So that led us to the hypothesis that the rate at which you apply your antimicrobial system will likely influence as well your treatment. If you have a slow rate, then you let the cell get the ability to adapt to the treatment you're trying to impose. Whereas if you apply it very fast, then it doesn't have the time to react and adapt. And we did this experiment with, and I don't show it here for sake of time, with irradiation, when you can apply the same dose fast and slow. And it's exactly what happened. When we apply the irradiation process at a slow pace, cells resist better because they get the time to adjust their metabolism and induce the bacterial stress response. Okay? So in the term of the 71, I mean, the temperature was applied very fast to get, at the core temperature, like what we do in the industry. But if you have a different pasteurization value with different temperature and rate, it can change totally the metabolism of the cell we're trying to control. When we did the microarray analysis, DNAK and GROW-EL were not in, the sets of genes that were found to be important to discriminate the different temperatures and pasteurization value. And at the time, we found that very hard. And if you've done microarray analysis, you have to do some cleaning of, of the spots, those that are overexposed and so on. And so we did some PCR analysis, real-time PCR, on those 2 genes to see if we were in control. And sure enough, they were so bright that those were among the genes that were eliminated during, the statistical analysis. But when we did the real-time PCR, you can see that DNAK and GROEL decrease as the severity of the heat treatment decrease. So basically, at the end, the treatment at 71 degrees Celsius is no longer significantly different than the control one. Whereas in the between, you can see that the cell was adapting. So now what we're trying to do is to find or to use the bacterial stress response as a mean to be able to establish when a process become or start to be effective. So it's no longer just looking at cell count, but what you've done, what it did to the metabolism, and be able to quantify it and determine it in terms of metabolism, not just cell count, because we know that if the cell are injured, they won't be able to form a colony. I just want, because I was asked to talk about meat, to talk about high pressure processing for a little while, because it's becoming very popular, but it's also very expensive. And because of the Listeria outbreak we had in luncheon meat and so on, this is one of the technology that became very popular. So the thing is with high pressure processing is that the food, or the meat in this case, is submitted to high pressure when it is in its final packaging. So you have the possibility of not contaminating your product because it's already in this package. So you just used high pressure using water, because you have an isostatic pressure transmitted by water on your product, and it affects the bacterial cells. So it's a post-packaging pasteurization. There's no post-processing contamination. Your product is already in its package. And you can get out for E. coli easily to the 5 log reduction we're asking for when you're pasteurizing a product. Okay? This is the kind of pressure you can use to obtain that pasteurization, but you have to be careful. At 600 megapascal, that can lead to some spore germination. So again, you have to know what is the microorganism contaminating your food. Does the pressure that you applied, what does it do? It's not because your product is already packaged that you're adding another antimicrobial system that you're not challenging what's in there. So again, you have to do a shelf life study after you've done the high pressure processing, although it's an addition of another processing controlling technique that you're using, because you never know what you induce, what is the metabolism, what effect you had on the cells in that. So again, even if you add another layer of security because you're putting high pressure processing, do a shelf life study to make sure that what you're trying to achieve, you do achieve. So in this case, prokaryotes are more resistant than eukaryotic cells, so it's easy to control, yeast and so on. And gram-positive are more resistant than gram-negative due to their thicker peptidoglycan cell wall. The mode of action of high pressure, the 4 hypotheses, or the things that have been, in the radar so far, is that it denatured or unfold proteins, including enzymes, so the enzyme is no longer functional. Modify cell membrane fluidity, so the exchange with the environment with nutrients is affected, proton motive force is affected, disintegration of ribosomes, so then they can no longer do their job. Intracellular pH change, it is very detrimental, either because enzymes are inactivated or because the membrane is damaged. So those are the 4 modes of action that are, linked to high pressure processing. What we know is that low water activity protect against high pressure processing. So food that have not so much homogeneous AW water activity, notably food with, emulsion with fat and oil cluster, is a challenge in this case when you're applying high pressure processing. To control spore formers, in order to sterilize the product, you have to add some heat. And right now, the technology is not yet there to be able to sterilize product effectively. We do pasteurize them well, it's working, but we still have to do some work to get to the sterilization process. Those technology are very expensive, and one way the industry here have been able to access the technology and not necessarily everybody acquiring the high processing pressure machine is we have now processing center. Of course, we do have major meat processor that now have high pressure processing system, pillars just to name this one. They were big enough to acquire it. But what do you do when your producer are small and they have, limited quantity that they produce? We have two now processing center, one in the western part of Quebec in Saint-Hyacinthe and one in the eastern part in La Pocatière. And what happens is that our processors do their job, do their product, do their processing, put them into their packages, and then before it gets into the grocery store or the distribution center, they go to that high pressure processing center to get the product treated, and then it goes on to the distribution system. So using those processing center, they do processing, high pressure processing, but various one depending on the food you have, and that has to be determined with them. But these center, that's what they do. They do high pressure processing of food. So the small processor have access to the technology. They just pay a fee for the quantity that they process. So that's how we've been able to get that technology that is still very expensive to those small producers, that have small volumes. So this brings me to my conclusion. I hope I've convinced you that nature is resilient, that the microorganisms are well equipped to adapt and resist, and not just to antibiotic. I like that picture when you see in one of our parks this tree just basically growing on just a piece of or a little bit of dirt. So that tells me that when we, wipe out nature, and that it's really that we have abused them. So nature is resilient. And we have to move towards a more global approach to what we do, and not just try to eliminate everything, but think about the health of the animal, our health, public health. We are in a way of changing the way we look at microorganisms and how we evolve with them. So that would be it, Enrique. So if there's any question, I'll be happy to answer them. Bon, là, c'est moi qui entends rien.
I will translate for you. ¿Preguntas para la doctora Linda? Las de la dignidad, aunque sea. Is the future in meat product sterilization to get rid of these persisters?
Well, the persistence is linked to antibiotics, so it's not a way of sterilizing them. It's just that as we give antibiotic to our animals, antibiotic resistance increase, and then we have it's a health issue, public health issue, that our animals, because we use antibiotic to treat them. We try to reduce the use of antibiotic as a growing factor to improve their growing performance when we produce them because of the health issue, public health issue. So that was just one more example to show you how microorganisms are resistant. We need to, they are well equipped to resist. We have to understand them well. So it's not a way of sterilizing it, it's just another example of how microorganisms are well equipped to adapt to anything we try to throw at them.
So it's more like we need to learn how to coexist with them, making use of this concomitant flora, like you said for listeria?
Yeah. you know, I know this all went very fast, but I have gave you Enrique papers that you can go through. The information that I gave is in those papers. You can, you know, take the time to look at them, and we'll be happy to answer any question through email if that doesn't come up right away right now. But the idea is really to have a new way of looking at the way we try to control microorganisms, better understand them, not just count them, and that wiping them out may not be the right way to go.
Ok, son las respuestas, ¿no? Para lo que dijo Diego. Muy bien, pues thank you very much, Linda, for your time. We really appreciate your contribution. And I guess you're free to follow up with your tasks of the late afternoon. Thank you very much. Gracias a la doctora Linda. We'll send you your certificates later on by mail. Thank you.
Wonderful. Bye for now.